Welcome everyone to week 2 of the fall 2021 Adirondack Research Consortium, or ARC, webinar series, Meeting the Demands of Climate Change. I'm Andrea Ferro, and I'm a professor of environmental engineering at Clarkson University and also a board member of the ARC. The ARC is a nonprofit organization that brings together researchers and decision makers to sustain a healthy Adirondack Park and growing vibrant regional economy. We're so glad that you're here with us today for this presentation. So I'm excited to introduce today's speakers to you who will be presenting field testing technologies to combat harmful algal blooms, or HABs, in New York. First, Dr. Lewis McCaffrey. He's a senior scientist, research scientist in the Finger Lakes Watershed program at the New York State DEC. He will be giving us an introduction to the topic of HABs. And then we'll discuss the HABs destruction technologies and demonstration projects with researchers from Clarkson University, as well as SUNY ESF. From Clarkson, we have Drs. Yang and Stefan Grimberg. Professor Yang is an assistant professor of environmental engineering who came to Clarkson 2 years ago from Caltech. And Dr. Stefan Grimberg, who's also professor of environmental engineering at Clarkson and the co-director of the New York State Center of Excellence in Healthy Water Solutions. And then finally, we have Ryan Lamastro, who's a graduate student in environmental resources engineering at SUNY ESF, and he'll describe the work that he's been doing with Dr. Stephen Shaw. These will be fairly short back-to-back presentations, 12 to 15 minutes, leaving time for the panel to respond to your questions at the end. We're also joined today by Dr. Melanie Johnson, who's the president, and Dan Fitz, the executive director of the ARC. So for your questions, please use the Q&A function to ask your questions. It's the tab at the bottom, and we'll get to as many as we can. Those we don't get to, we'll collect and provide answers on our website. You could also use the chat for greetings and other comments, but we'll be using the Q&A to be posing questions to the panel following the presentations. So I'm going to turn this right over to Lou to get us started. So Dr. McCaffrey.
Thanks very much. Can you hear me okay? All right, and you can see my screen?
It all looks great.
Okay, great. Thank you very much. Thank you very much, everyone, for joining us. I'm going to give an overview of harmful algal blooms to begin with, and then I'll talk about the mitigation strategies that have been used in New York State. And we're going to divide the mitigation strategies into HABs 1.0, which are more traditional methods of dealing with HABs, and HABs 2.0, which myself and the other couple of speakers will be talking about in more detail later on. So first of all, an introduction to HABs. What are they? Well, they are composed of algae, generally a broad term. Algae are single-celled plants that have chlorophyll and other pigments, and they use those pigments to conduct photosynthesis. So algae are present in all of the freshwater water bodies around the planet, apart from possibly the Antarctic. That's a special case. But they're they're omnipresent, and most of them do no harm at all. In fact, they're very important in terms of global oxygen cycling and carbon dioxide cycling. There's so many different kinds of algae. You can see some of the diversity there. But there are a subset, if you like, known as the blue-green algae or cyanobacteria. And although people call them blue-green algae, they're actually bacteria with special characteristics. They're highly specialized and competitive, and they've been around for billions of years. In fact, not far from where I'm sitting in Syracuse, we have fossilized colonies of cyanobacteria dating back to 400 million years. And there are examples of cyanobacteria colonies much older than that. So we know that they do best in high temperatures, and high light conditions. And they also are very good at taking advantage of high nutrients. The causes of blooms are not fully understood, although we're getting very close to really having a good understanding of them, I think. Despite that, they are hard to predict. We are expecting some blooms to pop up today and next week, probably due to low wind speeds, high solar insolation. So look out for those. So harmful algal blooms, or HABs. Well, let's just take apart that acronym. They're harmful for a number of reasons, because for a start, they can develop toxins. They decrease the economic value of lakes that they occur in. They are harmful to the aesthetics because we all like nice blue lakes. And ecologically, they outcompete the other algae species that are present. And once they die off, they can sink to the bottom of a lake and rob the water of oxygen, which harms the rest of the ecosystem. So as I mentioned, they're generally called algae, but they're not truly algae. And then blooms. There's various definitions of blooms. Some of them are based on the chlorophyll concentration, but effectively it's a proliferation of cells, a kind of an explosive proliferation. And we see them at the surface of lakes and rivers in very dense concentrations. So when do these occur? Well, they're typically around this time of the year, August to October. And you can see that a typical lake succession starts off with these dinoflagellates around May and June. Then we get a peak of the green algae. And then around this time of year, we start to see the cyanobacteria, and then back after October into the dinoflagellates. So I've stated that they are more common in lakes that have higher nutrients. The interesting thing is that they're also present in low nutrient water bodies too. And these include some of the finger lakes, which are very low in nutrients. They're known as oligotrophic lakes. And we understand that Lake Placid can also have some harmful algae blooms. We know that climate change is likely to have a significant impact on the proliferation or the presence of these HABs. And it does that through a variety of different reasons. One of the main ones being the warming of the lake water by the peak summer temperatures. For instance, our research has shown that New York State lakes on average are increasing in temperature by about 1 degree Fahrenheit per decade. Mussels, we think, also play a significant role, and that's because that mussels preferentially eat green algae and spit out undigested the blue-green algae, the cyanobacteria. And we also know that development and other land use changes play a role. So the common types of cyanobacteria that we see in New York State, on the left is Dolichospermum, also known previously as Anabina, and you can see that they contain these pigments that turn them green or blue-green, and Aphanezemenon, which is also another cyanobacteria, and you can see that they have these structures which allow them to fix nitrogen from the atmosphere, meaning that they can get one of the critical nutrients that they need. Microcystis is another common cyanobacteria. It forms these large colonies, and they are able to adjust their buoyancy so that they can move up and down through the water column. Now, they produce toxins. The ones on the left produce anatoxin, which is a nerve toxin, and Microcystis produces microcystin, which is a liver toxin. In addition to those microcystins and anatoxins, the outside of the cyanobacteria have endotoxins, which can produce skin irritation and allergic reactions. And that's the reason why we suggest that if you identify these blooms, not to enter the water don't swim in them, for instance. There are other more exotic toxins, for instance, cylindrospermopsin, which is more of a subtropical type toxin, saxitoxin, which is produced by marine blooms. But generally, we don't have to worry too much about them, these other exotic ones. They do have very complex molecular structures, and ESF is actually one of the best labs in the country for analyzing these toxins. So there's no visual clues that toxins are present. You can have a harmful algae bloom, which is completely free of toxins, but it can have toxins present, and we don't know based on what it looks like. So if you see a HAB, you can report it using these links. One is a link to a reporting tool, and this link, which ends with HAB form, allows you to report any HAB that you see. And I do suggest you go onto these NIHABs links and see where the blooms have been reported. Interestingly, there is a dearth of reports from the Adirondacks, and we think that it may be due to a number of different factors, which might not have anything to do with the presence or absence of HABs. It could be spotty cell phone reception, less density of people to report them, and so on. So I'm speaking to a really interesting audience here to find out what you think about that. Across time, we have seen an increase in the number of reported HABs in New York State. And the interesting thing is that the scientific community is still debating on whether there is an increase in phytoplankton or the cyanobacterial blooms, as suggested by Ho in 2019, or a paper that just came out 3 days ago, which talks about no evidence of widespread algal bloom intensification in hundreds of lakes. So the jury is still out. So that is a very brief introduction to HABs. Now about the state's activities. 2018, a lot of money was promised to deal with this issue, and some of that money went to mitigation pilot projects. So I'm going to very briefly introduce this. Now, HABs have been around for a long time. Humankind has had to deal with them for centuries, and some people would say millennia. And there are a number of commonly used strategies. One is making sure that the lakes are as clean as possible in the first place. That's nutrient reduction. So best management practices, improving infrastructure and erosion control and so on to keep sediment and nutrients out of the lakes in the first place. Once the nutrients are in these lakes, we can do something to manage the levels of bacteria or algae, chemical treatments and physical methods. So I'm going to go through those very briefly. So we call those traditional methods HABs 1.0, and we have tested them out in New York State under DEC sort of oversight. And so hydrogen peroxide was one of them that we looked at. It's known to work. It's infrequently used in New York State, but the algicide, hydrogen peroxide, breaks down eventually to water and oxygen. It's good at increasing the amount of oxygen in the water. We know that it probably shifts the algal community, and the fact that's what it's designed to do, but we wanted to check that. And it also may destroy toxins. We also tested out ultrasonic devices. These have got a mixed history of efficacy. They work by disrupting the flotation structures inside algae so that they're unable to move up and down the water column, and eventually they should sink to the bottom and die off because they're unable to photosynthesize. And we've also used nutrient inactivation, which is the use of chemicals to decrease the amount of phosphorus that goes up into the water column. This is definitely has been shown to work, but it is expensive. And we wanted to test a whole series of hypotheses to do with do blooms decrease, obviously? Are there changes to toxicity and chlorophyll concentration? And how significantly are the phytoplankton and zooplankton community structures altered? With nutrient inactivants, of course, we wanted to find out whether there is any reduction in nutrient concentrations. And in addition to the invertebrate community, does it get altered? So in order to do that, we collected an enormous amount of data. It wasn't my section that did this, but it was a very concentrated set of experiments that we did. And those results are really the subject of another talk, I think. But what we wanted to do was take the discussion further and find out what innovative solutions there could be to the HAB problem. And to that effect, we took part, or DEC hosted a workshop in 2019, where we looked at potential technologies beyond 2020. And these were some of the technologies which were suggested. There are some very science fiction sounding technologies here, including electrobeam radiation and the biological controls, including giving some of these algae and cyanobacteria viruses and phages. But eventually we decided on 3 different technologies. I'm going to briefly talk about one, and then I'll hand over to the other speakers to talk about the others. So the HABS 2.0 pilots, we started out with a smaller scale. We started out with rather a large scale system called Habitats. And this was put together by a contractor for the US Army Corps of Engineers. And the idea was to intercept algae on the lake, remove them from the water using air flotation, and then treat that sludge to turn it into biodiesel. And so we had that deployed on the side of a 64-acre water body downstate. And you can imagine that we've all seen these HABs, I think. Sometimes the HAB comes to you and sometimes it disappears. And that was one of the main problems with having a shore-mounted treatment system. It certainly worked, but what we wanted to do was try out a mobile version on Chautauqua Lake. That also ran into some engineering problems and weather-related problems. You can see some of the deployment images here. This was the background water quality, treated water quality. This was some of the sludge that was taken out. And this is some of the booms that were then funneling, hopefully, HABs towards this collection system. So that was the Army Corps of Engineers work. And I'm going to hand it over to the other speakers to describe their own work. Okay, I hope I squeezed everything in there as fast as I possibly could.
Thank you, Lou. That was a perfect introduction. I appreciate that. We're going to move right along to the Clarkson group, starting with Yang.
Hi, everyone. It's a great honor to be able to present our work done by Clarkson team. So let's move on. All right. So Lou has presented abundant information regarding how the harmful algal bloom happened. I just want to reiterate that harmful algal bloom did create a huge economic loss because of the impair of the visual damage on the water quality, and the concurrent release of cyanotoxin is really lethal to aquatic animals and humans. So it's a big problem, not only in the United States, but nationwide. So we are talking about treating a 100 acres, at least 100 acres of a big area of a water body. So that means conventional technology like batch reactor and beaker won't be able to accommodate these treatment needs. Therefore, we want to develop a highly efficient continuous mode continuous mode reactor to enable the treatment of large amount of water at a short residence time. To accommodate these needs, the key component we develop is the filter type electrode. So as you can see in the left panel, there's a filter electrode. We have synthesized it in a small scale and a large scale. So the porous, there are porous structure within this electrode, and we envision that if we do the pump and treat in this format, once the algae pass through the porous structure within the electrode, it will be instantly exposed to oxidants electrochemically generated within the pore, and then when the in the effluent, when the water cluster exits the microporous structure, cell will be inactivated, right? So this feature also give the system certain flexibility. So if we install it on a boat, we are able to move around the lake to enable the pump and treat. So we will talk about that in the following slide. Here, I would like to invite you to look at the electrode at a smaller scale. So this is the microscopic image of this of the electrode. There are cracks on this porous structure. That's actually the water channel. So as you can see in the left panel, so water will essentially flow through this water channel, and then algae will be exposed to these locally enriched oxidant produced electrochemically and then eventually exit the channel. We believe this design will significantly enhance the mass transfer coefficient. For example, if you just stir the water around the filter anode, the mass transfer coefficient is about 0.17. By the way, the higher the mass transfer coefficient indicate the higher efficiency for the algae to get in contact with the electrode and consequently exposed to the oxidant produced. So if we enable if we initiate the pump and treat process, as you can see, there is at least one order of magnitude increase, 2 orders of magnitude increase of the mass transfer coefficient. That means we are really we are really maximizing the mass transfer coefficient and hence promoting the treatment efficiency. So here are some test results. Surprisingly, we found that in the presence of chloride, the inactivation of cyanobacteria is significantly accelerated. So compared to the lab culture phosphate buffer solution, if in the presence of 1 millimolar or 3 millimolar of chloride concentration, the removal of chlorophyll A can really be accomplished within a region of minutes, right? At a residence time of minutes. So we use chlorophyll A as an index to quantify the viable cyanobacteria. So it turns out that the major oxidant in our system is actually chlorine, originated from the chloride in the water body. So chloride in water will be oxidized at the anode to chlorine, and then chlorine consequently inactivates the cyanobacteria. So the additional bonus of our system is that the chlorine produced is able to readily destroy microcystine. Here we choose microcystine LR as an example. In the presence of chloride, different concentration, we can achieve more than 90% removal of microcystine LR at a residence time less than 1 minute. So our conclusion is that the electrochemically produced chlorine not only inactivate cyanobacteria, but also enable the complete destruction of microcystine LR. So since we identify that chlorine is actually the oxidant, the audience, I can imagine there are questions raised in audience mind. why not just dump bleach like in the lake, right? So the bleach solution is essentially a chlorine solution. So we do an interesting comparison. Let's look at the blue line here. Blue line represents the electrochemical oxidation process. At 5-minute residence time, we are able to remove more than 90% of the chlorophyll A, which also refers to the cyanobacteria concentration. At this stage, we are producing 2.5 microgram per liter of free chlorine in the bulk solution. In contrast, if we decided to spike bleach into the lake water, we have dose 10 milligram per liter of free chlorine. Look at the black dashed line here to achieve the similarly fast degradation kinetics. So to summarize, electrochemical oxidation is more efficient compared to a chemical oxidation process and produce less residual chlorine in the water body. So we also do some fundamental study. So please ignore this equation. The main conclusion would be we are actually create a locally concentrated chlorine within the porous structure of the electrode. This is critical because the bacteria and microcystine will be instantly exposed to this concentrated, regionally concentrated chlorine. When they exit the porous structure, the concentrated chlorine will actually instantly dilute it in the bulk solution, leaving a very low residual concentration as 1.3 milligram per liter. So that also brings this feature also enable the production, minimization of the formation of disinfection byproduct. If we compare the chlorination, if we compare the electrochemical oxidation with the chlorination, see the orange bar and green bar here, a conclusion can be made that electrochemical oxidation produce lower amount of disinfection byproduct compared to chlorination at the same treatment kinetics. The x-axis shows the reference limit regarding the disinfection byproduct concentration, and the y-axis shows the ratio between the disinfection byproduct concentration versus the regulation guideline. 1 means the value below 1 means we are not exceeding the drinking water standard, right? So in order to facilitate the following field test, we decided to operate the system at a lower current density to make sure that our effluent quality, after the reaction can meet the even more stringent drinking water standard. So that's the effluent, the disinfection byproduct concentration, it will be lower than the drinking water guideline. So we are confident that this is a transformative technology. We demonstrate the proof of concept in the lab, and it is Professor Stefan Grimberg bring the whole design into reality. So the stage is yours.
Okay. So over the last couple summers, we then took those laboratory studies and scaled up our system to a pilot scale or demonstration system to demonstrate whether this would work also in the field. And so this is an image of our full-scale system where we have these electrodes on the these full-scale racks of electrodes on the side of the boat. It's a boat-mounted system, and then we have our power generator as well as pumps on top of the boat operating the system. Next slide, please. The efficiency, we showed that we can treat chlorophyll A. So on the left-hand side, you see chlorophyll A concentrations as a function of treatment capacity. And what you see is a lot of scatter, like what you would expect from typical data in the field, but you also see high removal. And then you see, obviously, as you increase treatment capacity, so the flow through the electrodes, you see diminishing removal percentages. But we did see high efficiency in terms of cytotoxin removal as well. We found that our specific efficiency of our system actually is more efficient in the field than relatively than in the laboratory work. So this is indicated by the specific charge as a function of removal efficiencies. So you see these red dots are relatively, we need less amps per hour liters in the field than we needed in the laboratory studies. The one problem we had mainly with our field work is that because of the filtered material, our algae actually clogged our electrodes eventually. So there was an engineering problem that we ultimately could not maintain the flow through our filter electrodes. And that diminished our treatment capacity at high flow rates. And that was our goal. We wanted to treat essentially a system about 100 gallons per minute, 100 million gallons per day. And so so what we tried to do then is developed another system, next slide please, which would mitigate or eliminate the potential that our filter electrodes would ever clog. And so in this system, we used essentially a plate configuration of our electrodes. Dr. Yang's lab came up with another coating material of catalysts on these electrodes, which reduced the energy requirement for the similar current density. So we ultimately had a 50% energy savings. And you see some laboratory results that we could treat the algae in a similar efficiency, or actually better efficiency. This electrode material also produced ozone in addition to chlorine as an active oxidant. And so on the right-hand side, you see our field reactor, which is for 100 gallons per minute flow rates designed. The next slide, please. So this, we tested that system on a large barge system. And so this is from our field work from last summer, where we ultimately placed the reactor. You see the reactor on the right-hand slide in operation. We also wanted to evaluate whether an extended residence time would help us improve our performance. And so that's why you see this white pipe there. So that was just a potential reaction chamber. And so we took samples before, so it's our control samples, in between the two, the electrode reactor and the post reactor, to determine the water quality. And quite visually, you see the slides, the bottles on the bottom there. So we see the green control water, so that's the lake water. After our treatment reactor or post reactor, we see relatively clean water already, and then even further treatment after the post reactor. But you almost see no difference between the two reactor statements. So we demonstrated that we could get 90% chlorophyll A removal after even after our first reactor at a flow rate of 100 gallons per minute in that system. Next. People always ask, when we ask about electrochemical oxidation processes, about energy consumption. And obviously this is a big concern when we deal with these large bodies of waters and trying to treat a lot of this water. And so the left part of that graph shows the average energy consumption, so kilowatt hours per cubic meter treated of our pilot system. And so we're treating our average energy consumption is about 1.1 or 1.2 kilowatt hours per cubic meter of water treated at a density of 7 milliamps per centimeter squared. So we can treat at our low current density with our specific electrodes at relatively low energy input this water. Overall, it will always require a lot of energy because we're treating a lot of water. I think there is no going around it, especially when we're dealing with larger blooms. Next slide. So what are we doing now with our technology? Dr. Yang and I started a company which is trying to market this technology, bring this technology to market. We're building a prototype with US manufactured electrodes, and then our goal is also to include a US supplied power supply on that system. So that will eliminate any supply chain issues, that we ultimately can produce these prototypes relatively quickly. We showed that the system is actually operated very easily, and our goal is to have those kind of mobile treatment processes available for small lakes associations who can ultimately mitigate their HABs outbreaks, HABs bloom, relatively quickly. Those are the people who would be there, who would see the HABs, and they could respond to these small blooms very quickly before even a large bloom would ultimately develop. And I think that would be our goal. I think the technology is easy to be handled that way. We also have funding from the National ICO program to identify more better our likely customers, so we can ultimately market that technology even better, and to help us ultimately identify the specific market for those kind of technologies. Next slide. And so with that, I think we are happy to move on to the next presentation from ESF. Thank you.
Thank you, Yang and Stefan. We'll move right on to Ryan Lamastro's presentation. Ryan, you can share your screen. Thank you.
Okay, there we go. See it all right?
Yes, looks great.
Yeah. This is me on video real quick, but my internet's a little unstable today, so I'm actually gonna keep it off. So we did, my advisor and I did research on cavitation of cyanobacteria as a treatment option for harmful algal blooms. My advisor being Steve Shaw and me, Ryan Lamastro. We had a great introduction on HABs, so let's jump right into what cavitation is. So cavitation is basically we're having flow through a pipe and then we force it through a small orifice. And in doing so, we have this exchange of pressure. We have a decrease in pressure and an increase in velocity. And that decrease in pressure, if we do it correctly, can drop the pressure below the vapor pressure. And when we get that, we start to have bubbles of water form. And later on, when that goes back above the vapor pressure, we have those bubbles collapse. And so what does this all mean in terms of harmful algal blooms? So we're talking about how it might damage the cyanobacteria. And cyanobacteria will have these gas vesicles that help to regulate their buoyancy so that they can go up or down in the water column. Some researchers think about the possibility that this cavitation, the collapsing of the bubbles, will help to disrupt the ability to regulate that. What it also might do is we might start breaking apart colonies. So colony size will help regulate where cyanobacteria are in the water column. So right here, we can kind of see we have larger colonies and smaller colonies within an image. And this, so why does colony size matter? So if you imagine you take like a handful of soil, throw it in the water, you'll see the larger particles like sand and whatnot, they'll settle fairly quickly. But then the smaller particles like clay and whatnot, they might take quite a while to settle, or they might even just stay in there indefinitely. Now we can flip this around for buoyancy. A larger particle that has buoyancy will be able to rise faster, whereas a smaller one might take much longer or maybe not even reach the surface. And this goes into cyanobacteria possibly being able to outcompete other stuff like green algae and other things in the ecosystem, and then that they will take all the incoming solar radiation energy and kind of leave none for the rest. And so what does this look like? Here we kind of have what our field setup looks like. It is a pump, and we have that basically is attached to a frame so that we can rest it on floats. And that pump is inside an inlet that allows water to flow over the inlet into the system. And then it'll pump it out through the orifice plate up there, which is where all the cavitation occurs. And then we just have a pressure gauge to regulate, to keep track of the pressure as it goes on. And what we do in the lab, or lab experiment design, is we take some water from the lake in 4 containers. And then from each of those containers, we take half that water and we cavitate it and put it in a column. And then we take the other half and we run it through the pump, and that acts as our control. We run it through the pump without the cavitation plate. And then what we see at the bottom here is that we'll see not too much of a layer form at the top of the column for the cavitated, and we'll see a lot bigger concentration of cyanos at the bottom. And then for the pump one, we'll see kind of a layer form at the top, while some of it, some but not as much, sink to the bottom. And so what potential advantages the system has on a near shore? So we can use a shoreline power source. So we can pretty much hook it up to a dock, run a wire, and put it in there. We don't need a boat and we don't need an operator. This can run pretty continuously. For lab work, it means we basically just had to sit out there for hours while it ran. And it can be scaled by adding more units. If you wanted to cover more coastline, then you would basically just put in another unit. And it's relatively safe. The main thing that it consists of is a well pump at this point, which is not terribly harmful. And here we kind of just have an example, one of our in-lab experiments, where you see on the left, we have the cavitated, and the top part is relatively clear. And then you can see kind of a green film at the bottom of all of the algae settling out. And then on the right, we have our control, or our non-cavitated one, where it has that big film at the top and then not so much at the bottom. We kind of have we've also done this in volumetric flasks, where you can kind of see it even better. You just have those little lines where in the control, it forms at the top, and then in the cavitated, most of it settles out to the bottom and you don't see too much at the top. So what we're trying to do is more explain the settling at this point. So here are some images. On the left, we kind of have a typical image that we would take for a cavitated at the top. And what we see is there's really not too many colonies and they're relatively small. Whereas on our control, we have quite a bit more colonies and they're quite a bit larger. So right now, what we're working on is actually quantifying the colony size in all these images to see if there's any discernible change. And what we're also starting to look at is there a dominant species after we do this? Do we see more microcystis versus other algae in the top portion of the column after cavitating? So like, would microcystis maybe be more resistant to cavitation or more vulnerable to cavitation as opposed to other species? So to date, some of our main conclusions is that we definitely see a change in stratification as for high concentration scums. Now, when we have smaller concentration scums, or not scums, but when we have smaller concentrations, we don't see as much vertical effect on the vertical movement within the column. So what our next steps generally would be is to try to test this on a field site that we would be able to have a more consistent surface scum or high concentration on the top of the water. Because right now our main field experiments were only on lower concentrations in the water column. And that's all I have.
Thank you so much, Ryan. And thank you to all the presenters for your talks. So it was great to get the background and then the technologies and field demonstrations and results from those. So thank you. For anyone who has questions, feel free to use the Q&A. Professor Grimberg and Yang and well, everybody's been responding to questions during the presentation, but so let's go to an open question that that Stefan just responded to. But at what depth, hang on, does the barge have to be stationary during operation, or can it drive around while operating? So that's, this is for the Clarkson group. So let's talk just in general about the operation and limitations or advantages of the design.
So I guess I'm happy to answer those questions. So the barge, the intake from the barge for that treatment process can be adjusted, and we could adjust it where obviously we want to take in the most contaminated or algae dense water for treatment. And so we tested at about a depth of about between 1.5 and 2 feet below the surface of the water, because that would be the water people would be most concerned about. I mean, I think the floating, if there's algae floating below that surface water, they wouldn't notice it as much, for example. But that could be adjusted. It's just an intake hose with a screen. In terms of whether the barge can move, we tested it stationary, but there's no reason why this. We essentially envisioned that the system will be on a pontoon boat on a smaller scale, so we don't have to have a barge, and it could be moving while treating. So I think that's the advantage of a mobile system, that we actually can move it around.
Thanks.
Mm-hmm.
We have another question on the, do you predict any variables in field experiments that might affect the effectiveness of cavitation? So this question is for Ryan.
Yeah, I think our main concerns in a field experiment would be basically keeping the pump running appropriately, because if the pump ends up providing a lower pressure or whatnot, the cavitation will then not occur or not be as good. So basically having a pump that would be able to run for, say, 8 hours without letting up on pressure, or basically being able to make sure that that pump doesn't get inhibited by any other stuff in the water. We can't just dump the pump in the lake and hope that nothing gets in it. So that's our main concerns in the field right now.
Thank you. And then are the procedures for limiting all algae or primarily cyanobacteria, which we found out from Lou's not algae after all. Does anyone want to take that?
I guess I can take that. So I think our process of electrochemical oxidation is not selective. So it actually takes any organism that enters the reactor, and it will kill or oxidize that because of the presence of the high oxygen concentration locally. So it would ultimately eliminate all kinds of algae or cyanobacteria at the same fashion, which actually might be a concern by some other people who would say, well, why don't we want to just remove the problem organisms like the cyanobacteria who ultimately produce the toxins. So I think that's a double-edged sword. So I think these methods are not selective. I think at least our method is not.
Thank you. And I saw that in your response to a previous question, you talked about how, you're focusing on the area with the algal bloom
Right.
or cyanobacteria. So you're
Right.
limiting the effect. Great. So there's a question on effectiveness of barley bags. Is anyone familiar with using barley bags and the effectiveness of that?
I'm going to send that to Lou.
Yeah, Lou hasn't spoken in a
Come
while.
on, Lou. I think this is yours.
Yeah, thanks very much for that, Stefan. We haven't done any experiments on barley bags, so I know that it's kind of almost like a folk remedy, but I can't really say one way or the other whether it's effective or not.
Thank you. Lou, while you have your camera on, I'd love to go back to an early question, the first question that came in on the effect on wildlife. I think all four of you touched on this in some ways, but maybe you could make a comment on that.
Yeah, sure. So we in DEC have been very interested in finding out whether any of these methods do affect wildlife, fish larvae, fish eggs, and other parts of the ecosystem. So we were very active this year in supporting the Clarkson project on Lake Neartowanta near Fulton. And we collected samples for analysis by the Cornell Biological Field Station. That was mostly for zooplankton. And we also took samples to test whole effluent toxicity, which means how toxic generally is the effluent that comes out of Clarkson's technology in this case. And the results are still being analyzed, but generally we found no major effect or no effect at all actually on the test organism, which was minnows. And we did see some effect on Daphnia, which are, a zooplankton, but it's still to be calculated whether that is a statistically significant effect. And interestingly, the lake water from Lake Neartowanta also showed some toxicity to Daphnia. So we're still analyzing the results, but it's a very valid question.
Thank you. Does anyone else want to comment on that question? I saw that Stefan responded in writing on the chlorine, and Yang talked about the chlorine meeting the drinking water standards from the effluent. Okay, great. There's another question that came in on the volume of the treatment, or the volumetric flow rate through the treatment. So it seems fairly small, and I see Stefan nodding, so maybe you can comment on that.
Yeah, so these are small systems that are never intended to treat an entire lake per se. So I think we were asked to develop a technology as an essential emergence response to an emerging small bloom or shoreline bloom. So these are the blooms which happen sporadically. They show up very quickly and then they disappear depending on wind conditions or hydraulic conditions. But they will cause beach closures and cause essentially economic discern essentially because of the beach closures or we did essentially the toxicity evaluations. And so we're looking at a process which we can ultimately clean up a surface bloom, a small surface bloom within a couple of days of treatment. And so that's where we, if you assume that essentially the bloom is on the first couple of feet of a water body, and you treat at 100 gallons per minute flow rate, you can actually treat a substantial amount of area in significant amount of in a relatively short amount of time, in a couple of days. Again, this will never treat the entire lake, or it will never eliminate all the conditions why we have blooms to begin with. That's when you need these mitigations, what Lou was referring to, of removing nutrients, discharges into the lake and trying to essentially have larger scale mitigation technologies. But that costs a lot more money. So I think this is really in between, an in-between solution where we trying to keep a beach open or keep essentially reduce the toxicity of the water.
Thank you. Yeah. Ryan, did you have anything to add to that from your system?
So my system would largely be a near shore system. So it would maybe be a system to kind of try to clear up a dock area or whatnot. It's not nearly as mobile. We don't have it on a barge. We can't really bring it around the lake as much. So it would kind of clear up a dock. And if you wanted to clear up another dock, you'd probably need another system because our flow rate is much lower than that even. We're probably more on a scale of 1 to 10 gallons per minute.
Okay, great. So you're looking at potentially the same beach closure, smaller scale where you're targeting your treatment.
Yeah, and we try to treat like the top inch even of water. So
okay,
on the same thought, clearing up the top is not as much of a volume.
Right. I wanted to go back to Lou's comment in the beginning on so recognizing where these algal blooms are. So you showed the map and where they've been reported. And I was just wondering about technologies for, so this is people reporting in to the DEC website, but I'm wondering if there are technologies for discovering the blooms, without people present, for example, satellite imagery or anything like that.
Oh, it's almost like I fed you that question, but I promise I didn't. So
Nope.
I'm working with SUNY ESF on a project to look at satellite imagery using the European Space Agency's recently launched Sentinel-2 satellites, which are relatively high resolution, and it should image around 8,000 of New York State's lakes every couple of days. And we're developing algorithms which should then be able to estimate the amount of chlorophyll in the surface waters, which will give us a very good idea of the location of blooms. So yeah, we're definitely very active on that. DEC is funding that project. We're also working with USGS on an advanced monitoring project, and that has been going for the last 2 years, looking at very sophisticated technology that is moored or anchored in the lake, and it's able to tell us information about nutrient concentrations and also phytoplankton concentrations. Particularly, it's able to tell us about whether there are cyanobacteria in the water. So that's more of a local, but more high resolution, more high fidelity system. So there is a lot of work going on this subject. Thanks for the question.
Terrific. Yeah, thank you. Well, we're sort of running out of time here. Oh, there's one more question that came in. What methods of treatment would be the most effective near tributaries? So we'll call this our final question. And would anyone like to respond to that? Yang, we haven't heard from you in a while.
Well, if you ask me about the best practice, I would say electrochemical oxidation, right? So I believe it's difficult to answer the question before we evaluate. We executive a full in-depth evaluation of all the technology. So all the technology have their niche. I know the limitation of mine, so I won't tell it here. So, but I think it's too early to answer that question. Yeah.
Okay, thanks. Any final comments from the panel? Okay, well, for finishing up right on time, I would like to thank everybody for joining us today, the panelists, the participants, and there are a few more questions that are coming in, so we'll collect those and post answers on the website and with the YouTube. So this will be available via the website. There'll be a link to the YouTube for the presentation. I'd also like to thank our sponsors. So support for the webinar today was provided by the Glenn and Carol Pearsall Adirondack Foundation, Paul Smith's College, Cloud Splitter Foundation, the Lake Champlain Basin Program, the New England Interstate Water Pollution Control Commission, and International Paper. So thank you to the sponsors, thank you to the speakers, and to the audience. It's been great. Hope you join us next week. We'll be meeting the panel chair with Kelly Serralo, talking about bioregional collaboration to build social, economic, and ecological resilience. Take care, everyone.